Welcome to the C2 Pipeline Innovation and Curiosity Center Chromatography Lab. Chromatography is a word derived from the Greek word chroma, meaning color. Chromatography provides a way to identify unknown compounds and separate mixtures. Chromatography is a method of physically separating mixtures of gases, liquids, or dissolved substances. Chromatography can be used to identify drugs, poisons, and many other substances. Separation is determined by the molecular size and/or charge. Mixtures and compounds. A mixture is two or more substances that are mixed together but not chemically combined. Examples of mixtures are soda pop, which is a mixture of soda syrup, water, and CO2 gas, air, which is a mixture of gases, and Kool-Aid, which is a mixture of water, sugar, and flavor crystals. Compounds are two or more elements that are chemically combined. Some examples of compounds are salt, which is sodium and chlorine combined chemically, carbon dioxide, which is carbon and oxygen combined chemically, and water, which is hydrogen and oxygen combined chemically. Chromatography is a technique for separating mixtures of compounds, identifying unknown compounds, establishing the purity or concentration of compounds, and monitoring product formation in the pharmaceutical and biotechnology industries. It is widely used by forensic teams to analyze blood and urine samples for drugs, for pain analysis, and for testing the presence of explosives. All forms of chromatography involve two phases: the stationary or absorbent phase, which includes the material on which the separation takes place, and the substance to be separated. And the second phase, the mobile phase, where the solvent transports the sample into different colors via capillary action. There are 5 types of chromatography, as seen here. Most chromatography uses modern instrumentation and involves placing the sample to be analyzed on a support, paper or silica, and transporting it along a mobile phase. The mobile phase can be a liquid, which is liquid chromatography, or a gas, which is gas chromatography. Chromatography applications for the matching of ink types includes HPLC, or high-performance liquid chromatography, which is usually done at a lab, TLC, or thin layer chromatography, and paper chromatography, which we will perform today. Let's learn about some of the different types of chromatography. In gas chromatography, a gas separates the components and then they are ionized, and an electrical signal is recorded in the mobile phase. In high-performance liquid chromatography, this is done at room temperature, which is advantageous for chemicals that are heat-sensitive or volatile. A liquid, which is the mobile phase, is pumped through a column containing fine particles, which is the stationary phase. Thin layer chromatography is when a plate is coated with a granular gel, usually silica gel or aluminum oxide, for the stationary phase. The substance to be separated is carried up the plate by capillary action. The substance with the most affinity or attraction for the plate will rise the farthest. Paper chromatography is the same as thin layer chromatography, but paper is used as the stationary phase. Electrophoresis, sometimes called column chromatography, is similar to thin layer chromatography, but the substance is separated through a gel by electrical current. Due to different size and charge, substances will move across the plate at different speeds. Chromatography as evidence. Class characteristics may include general types of pens, pencils, or paper. We will look at this today. Individual characteristics may include individual handwriting characteristics, trash marks from copiers, or printer serial numbers. The next part of this movie will show us how to set up our filter paper. As we go through the steps to set up the filter paper, please do not perform any of these steps until the movie has been paused and further directions have been given. This will avoid errors. Our first step is to look at the filter paper. The bottom of the filter paper, flat or straight, cut across. The top of the filter paper is rounded, as shown in the picture. Our first step will be to use our pinky finger or smallest finger, place it at the bottom or straight edge of the filter paper, and draw a line using pencil above your finger, just as it's shown in this picture. We will pause now to complete this step. Our next step will be to write under the line, first the letter N in the very left corner of the filter paper. Then, in the very right corner of the filter paper, we'll write the number 3. Next, in between the N and the 3, we will write the numbers 1 and 2, spacing them evenly as shown in the picture. So when you look at this, the N is in the far left corner, the 3 is in the far right corner, and the 1 and 2 are in the middle, and they're all separated with even distance between the letters and the numbers. We'll pause the video to complete this step now. Our next step is to use the marker labeled number 1, using the very tip of the marker to make a small dot. You'll just tap the tip above the line above the number 1 as shown. Do not draw a circle. You simply need to tap the tip of the pen above the number 1. We'll pause to perform this step now. The next step, we'll use the marker labeled number 2. Again, using just the tip of the marker, make a small dot above the line above the number 2 as shown. Remember, just tap the marker above the line, above the number 2. We'll pause to perform this step now. I bet you can guess our next step. Our next step will be to use the marker labeled number 3 and use the tip of the marker to make a small dot above the line above the number 3 as shown. We'll pause to make sure everyone has completed these steps. We are now ready to begin our lab.